Imagine if the key to healthy aging wasn't just about avoiding disease, but about understanding how your immune system silently shapes how you age at a cellular level. What if the inflammatory footprints embedded in your biology could predict and even be shifted to improve your late years? Today's guest has been unraveling exactly these questions. Welcome back to Decoding Longevity. I'm your host, Raghav Sehgal, joined as always by my co-host, Macsue.
Hi everyone. In our last episode, we have explored how cities, communities, and clinics are becoming the new test beds for longevity research, and how scientists are starting to find ways to measure effects of joy in health. Today, we dive into a little bit of a different topic, but equally transformative. So, the immune system's role in aging and how chronic inflammation and microbial interplay shape our biological age. Our guest is Dr. Alan Landay, professor of internal medicine and immunology, whose work has reshaped how scientists think about immune activation, inflammation, and aging, particularly in people living with HIV, but with implications that reach far beyond any disease. Alan, welcome to the podcast.
Well, thank you. It's really a pleasure to be here today and really talk about this very exciting topic that really has tremendous global implications in understanding aging and the basic mechanisms and underpinnings, and really bringing what I have of over 40 years of experience in the HIV field to now understanding aging and applying it to the global population.
Absolutely, Alan. And actually, that's kind of where I want to start. Your research has really brought attention to sort of how chronic and innate immune activation, also sort of coin this term of inflammaging, contributes to age-related decline. So can you kind of unpack for us what that means for our listeners? And how did sort of the studies from your HIV research, which has been really global in a way, reveal insights into aging biology specifically?
Absolutely. It's been a very long journey, and starting as a postdoc at the University of Alabama, Birmingham, I studied some of the first subjects with hemophilia and a new disease that we weren't even defining yet because 1981 and 82, we didn't know about the virus that was causing this new disease. So it was an immune deficiency disease. We knew T cells were depleted, but we did know even back then about immune activation, and we're able to characterize that with some of the newer developed monoclonal antibodies. And monoclonal antibodies and flow cytometry at that time were very new tools in the field of biology and science and immunology. So it was at the very beginning. We then studied this further, and I moved to Chicago to take on a position there and really began to study in earnest this new disease of HIV and began to see how inflammation was a big part of the underpinnings of the biology and pathogenesis. And even when we were able to treat the disease and actually get the virus under control, reconstitute the immune system, all that happened not only in 1995. So it was only 30 years ago that we really saw a revolution in HIV care and therapy. People, when I studied hemophiliacs, they were dying within a month of their diagnosis. But in 1995, people were living longer. But we still recognize that we were now controlling the virus, but the inflammation that was present in the blood of these subjects still persisted. And we could see, if we fast forward about 10 years, that these individuals living with this persistent inflammation now were getting diseases of aging at a much earlier age. So in the mid 2000s, we were able to see now cardiovascular disease, neurocognitive decline, metabolic diseases in people who were only in their 50s, where you'd normally see this in people who were in their 70s. So HIV, because of this persistent inflammation, was really a result of the ongoing potentially virus or other dysregulation of the immune system at this time that really led to these other diseases of aging or non-communicable diseases.
Yeah, it's a great intersection to see that how HIV was actually accelerating aging and accelerating the biology. So I guess when we talk about aging, it has become very prominent the use of clocks and biological clocks, which sort of like signifies a different age than the one that you actually see in your birth certificate. But again, going back to the inflammation, there's a whole involvement of the immune system. So what have you found, and what does the immune system have actually to do with aging itself?
Well, that's very critical. The immune system, and it's really, we now believe it's the innate immune system, the monocytes, and at the tissue level, the macrophages. So in your brain, you have monocytes that become microglial cells, and they live in your brain, and they can produce inflammatory mediators or inflammatory metabolites, and that's due to their activation status in the periphery. We can also study those same cells in your liver, called Kupffer cells, or in your lungs, alveolar macrophages. So these immune cells are very critical for making these inflammatory mediators that then are pervasive throughout the body and throughout all of your organs. One of the critical questions is, what were the drivers of this inflammation? Why did it persist? Well, we were able to first identify that back some 20 years ago in studies that we collaborated on with the NIH, and found that it was due to a leaky gut, that HIV, when it infects a person, no matter what route of acquisition, goes and homes to the gut. And why is it? It's really related to what Willie Sutton was asked when he was asked as a bank robber, why do you rob banks? Because that's where the money is. And again, why does HIV go to the gut? Because that's where its target cells are, CD4 cells. So once those cells are infected in the gut, you then alter the gut microbiome, alter the gut epithelial barrier, and the products of the gut, these innate immune stimulators, get out into the blood and activate these innate immune cells and cause them to make inflammatory mediators, as I said, and that can happen throughout the body. And this now persists even if we treat people in the earliest stages of HIV during their acute infection. The leaky gut is not repaired even if we give them these very potent drugs that we have today. So that's a very consistent finding, and one that sort of is now what we know is the importance of the innate immune system in driving persistent inflammation, leading to the disease of aging.
And sort of this gut dysbiosis is more and more becoming a really strong sign of dysregulated aging, a hallmark of aging, I would argue almost. And sort of your work around sort of this multi-omic signatures of aging in gut dysbiosis and its relationship with cardiovascular comorbidities is also really interesting, suggesting potentially sort of this relationship with microbiome just not being a passenger in aging, but potentially being a really active player. Could you talk a little more about this, that how sort of gut dysbiosis might be really playing a really critical role here, especially in cardiovascular aging?
Absolutely. What we're now finding, and we've been studying this in very large cohorts, there is a large cohorts in the United States called the Macsue cohorts.
Hmm.
They've been in existence for over 40 years for the Macs and over 30 years for the Wise cohorts, both men and women infected with HIV. And we also, beauty of these cohorts, they have HIV negative match controls for both men and women. So it really allows us to match very carefully on all the characteristics of these individuals except for HIV. We now have very large NIH grants where we're looking at the microbiome and those microbiome changes in the guts of these HIV infected men and women compared to their healthy controls, and finding that several of the bacteria that are altered are now linked to cardiovascular disease outcome. And many of these, and the reason why, is that we lose bacteria that make the anti-inflammatory short-chain fatty acids. And that's what maintains the low inflammation in a healthy donor. But then in a donor with HIV, we're driving bacteria that do not make the short-chain fatty acids. And it's that correlation of the short-chain fatty acid in the blood to cardiovascular disease. And those seem to be very protective. So when they're there, they can protect against the inflammatory diseases like cardiovascular disease. And when they're lost, then you develop more cardiovascular disease. So we're finding HIV causes the dysbiosis, the changes in the bacteria, the loss of short chain fatty acids, and this is now linked to what we see in terms of cardiovascular disease outcomes in these cohorts.
Yeah, it's very interesting. Actually, someone, a colleague of mine here at Monash University, she's heavily working with hypertension. And, one of the things that she does look is the gut microbiome and looking how the lack of fiber actually also increases the risk, or actually can be also almost like a causal factor for developing hypertension, which is known, it's highly linked with cardiovascular disease. So it's really, really interesting, this whole role of the gut microbiome playing a large part in multiple diseases from HIV to cardiovascular and so on. So I know that your career started heavily on the HIV, and we spoke about it, offline on this, but now has completely shifted into more of the aging independent of the HIV. But I guess there is a lot that can be translated in understanding aging for a general population. So do you mind talking a little bit more about this shift, and how do you see now all of this coming together to really understand aging as a general for everyone, not the ones affected with disease?
Well, exactly. So almost 15 years ago, we began to look at the idea of HIV coming together and thinking about it as a disease of aging. We had good drugs, we were controlling the virus, people were getting these diseases, cardiovascular, metabolic, neurocognitive, and even some of the cancers were coming up. And we then realized that this was a accelerated, accentuated aging, and that we could then move what we learned in HIV and apply are these same inflammatory mediators present in healthy donors, but at an older age, maybe at 60s or at their 70s, where we're seeing it in the 50s. And we did see that. What about gut translocation? Well, we started those studies some 15 years ago and found that in a 70-year-old looks just like a 70-year-old HIV negative looks like a 50-year-old HIV positive in terms of these inflammation changes and leaky gut. There were actually studies in 1995 looking at telomere changes, and that was very early on, and comparing the fact that an HIV person at age 60 had telomeres like that of a 100 or 80-year-old. So there are all these findings of accelerated aging, accelerated disease that we could use to now apply in the healthy aging population and see what happens there, and try to use some of this information to now apply to HIV, especially where a lot of the immunology comes from. We now know about the effector cells like CD8 cells, we know about CD4 cells, we know about natural killer cells, we know about the role of monocyte macrophages. There are many parts to the immune system, regulatory cells, and so on. And a lot of this immunology we learned early on, almost 30 years ago in HIV. We now learned actually early on in HIV about immune senescence. Now that's something the aging field took on much later, but we started looking at immune senescence actually back in the 80s. And colleagues of mine were looking at markers on their immune cells in HIV showing, hey, these cells are senescent. They could show telomeres were lower. They were actually making inflammatory mediators. They were becoming senescent, sort of the zombie cells that are still alive but making inflammatory mediators. This was before people knew about senescent associated secretory phenotype, because those individuals were not studying the immune system, they were studying the tissues during the aging process, like endothelial cells or adipose tissue. So a lot of the early immunology taught us to now look in aging individuals who don't have HIV, and now we're finding all the same features. We knew early on that much of this in individuals with HIV also may be driven by another virus, CMV. And CMV came up early on in our studies of HIV as a co-infection. Almost 100% of our HIV patients are CMV co-infected. And we now find that it's that virus that drives the senescence. And what are we doing today? Actually, today we're trying to treat, now that we can control HIV, CMV may be the factor driving this persistent inflammation. So we're now starting to treat our HIV patients with CMV drugs, but we have to move that into healthy donors. So we're learning a lot about therapy, approaches of pathogenesis that we can ultimately translate to the rest of the aging population of the world.
That actually is an excellent segue into therapy. Now we've been talking about biomarkers and potential indicators of immune aging. Why not let's talk a little bit about therapies and interventions, right? Um, what do you think and what do you see as the most promising sort of interventions and therapies that we can see for slowing down unhealthy aging? Are they going to be lifestyle based? Are they going to be microbiome based? Are they going to be immunomodulating or something else altogether? What are you going to be placing your bet?
Well, the one thing that excites me, and we're opening a study here, are the GLP-1 agonists. They're getting a lot of interest. They're currently injection based, but there is a newly licensed GLP-1 agonist that is oral,
Yeah.
and there's going to be more of those formulations available in the next month. So I think it's going to be easier to get the drugs out. Now, that's great for a drug, but one of the things we start talking about, and I was on a call this week with the World Health Organization discussing this about where are they going? They're interested in neuroscience. They're interested in this new field. But when you move to resource limited settings, and I don't want to leave out that part of the world, we have to look at lifestyle. It's going to be diet. It's going to be exercise. How do we look at that and control those opportunities? I think we can move a lot of these drugs there as they become more accessible, but we also have to think about standard approaches like good exercise and good diet. But the GLP-1s are very exciting. We're starting a low-dose tirzepatide trial here in
Amazing.
Texas, and that will be kicked off, I hope, by next week, first patients. And what are we going to use? Our endpoint outcomes are going to be the aging clocks. In this case,
Wow.
the third generation clocks are what we're going to use as our outcome measure. So what we want to do is start with currently licensed drugs. So it's much easier to get started. You don't have to file an IND with the FDA. We're actually using the lowest dose of the drug that's on the label. And so we're not using it to look at weight loss, but to affect health span and healthy aging. And
Amazing.
that's just the first of many things that we want to do is to look at those drugs together, maybe rapamycin,
Yeah.
maybe SGLT2 inhibitors.
Mm.
All of those are licensed for a lot of our current chronic diseases, and we think those also might have an impact on health span.
Yeah.
We're also very interested in actually taking something from our HIV playbook, a reverse transcriptase inhibitor that can affect the human endogenous retroviruses and line elements. And
Yeah.
we just looks like we'll be funded soon to do a ARPA-H trial to work
Wow.
on that. And so
Amazing.
that will give us a great opportunity to test the hypothesis of how about if we use the drugs that block endogenous reverse transcriptase, the same drug that was being used to treat exogenous retroviruses, HIV. So again, taking something from our HIV playbook, we now apply it to the healthy donors to see is that going to affect brain aging or is
Yeah.
that going to affect cardiovascular aging? And I think it's a very exciting time. Now, we're going to go beyond that. We're certainly going to look at new opportunities and we're going to try to target maybe things like CAR T cells in the future might be a new bold area and some of the repurposing drugs and looking at all the ways in which we can rejuvenate the immune system and bring it back. But we have to start very simple and very focused and try to start where we can really have some impact and then go forward from there.
Yeah, I hope you're also looking at combination of drugs because I think that's such a big thing that it's so understudied. Like we don't even know some people are taking so many things at the same time, and there is really no research because we tend to look at okay, what is the effect of this one single thing at a time? But I think combination of drugs it's highly important. So I hope you have space to do some of this as well.
Absolutely. And what we want to do is use AI. I've
Mm-hmm.
just had that discussion with our AI team here at UTMB. And we really want to do this in a very informed and methodical way and very make it so we could really have an impact. And I can't agree more that what we haven't talked about are the 12 pillars of aging related to the geroscience hypothesis, which really gets us to targeting things like mitochondrial inflammation, dysbiosis, autophagy, looking at DNA methylation, and so on. So I think we have to take each of those pillars. I was just having a conversation earlier today with a colleague, and we were saying we need to understand the phenotype of our patients. Which part of these pillars is that person most impacted by? And if we're going to target a pillar, and we have the assays, and we have to have good assays. We have to use the newer therapies, but we have the good, solid assays. And ultimately, the outcomes are going to have to be such that what's going to be accepted by the FDA.
Yeah.
Now, the trials we're doing with ARPA-H, we're going to use a functional measure of intrinsic capacity. And that's what's going to be used overall as an overall functional measure with the 5 domains for intrinsic capacity. But we also want to understand more about the biology in those studies. So we will do biologic aging clocks, we'll do proteomics, we'll do metabolomics, glycomics, and all of this will come together in a multi-system approach to understand mechanistically what's there. But it's a real brave new world where we're trying to head.
Yeah, it's incredible. I guess like it is very clear that aging is not just about time, but the state of our biology as a whole, and looking at all of those pillars, all the hallmarks of aging and how all interacts together, right? So before we wrap up, Alan, I guess my last question is, what is coming up in the future? We spoke about a new clinic or a new institute. So what kind of like discoveries and collaborations like are you getting involved and keeping you, excited in the
What
field?
excites me is locally we want to develop a whole geroscience institute, and we really want to make it from the bench to the bedside of the community. We're on a small island, so we have the great opportunity. We're going to form a new cardiovascular heart study for the island, and that will involve 5,000 individuals. So that'll be a great opportunity to look at these individuals, what happens to them as they age. But it's not just aging in your 60s or 70s, it's aging across the lifespan. And I think that's another thing when we talk about health span versus lifespan, but it's also when does aging begin? Well, it begins at potentially conception. So studying pregnant women is as important as studying an 80-year-old or a 70-year-old in understanding aging, because what you're exposed to in your womb as an unborn child may affect you when you're 70 or 80. And now we're also looking at the environment. So we have a whole new program we're building on exposome and the environment. I live in Texas. There are oil tankers coming in right now into the harbor as I look out my window across the Gulf. And there are oil refineries literally a mile down the road from me. So this is going to affect us. But we're also excited to look more broadly at how can we study aging in a much more out-of-the-box idea. So I was discussing today, what about the oral cavity? We just simply use and sample the oral cavity. How does that affect aging? What happens to skin? Can we all look different as we age? Can we use and study skin as an aging outcome measure? And of course, other aspects of the microbiome and functional measures. So we're really trying to do this as a truly integrated view of science in this new geroscience lab. Use all these newer tools, use these existing drugs, and eventually give us some time to develop newer drugs that we can apply and eventually change the way we live, because we all want to be 100 years old out running down the street, or as I do, play pickleball on weekends. So I'm 70, so at 100 I want to be still going to that same gym, playing pickleball or lifting my weights and my kettlebells that I do every morning. So that's what I'm looking for, is a good health span for myself, and hopefully all of these things that we're working on will help contribute to that.
I can tell you, Alan, you don't look a day over 50. Uh, well, Alan, thank you so much for joining us today and sharing your amazing, amazing insights. This conversation has been so insightful and really went into the deep dives of the immune system. But you told us a lot about how the immune system can be a central driver of aging itself, and we really need to focus more on it. And for our listeners, we'll link a lot about Alan's work at UTMB and beyond, along with his other interviews and aging resources in the show notes. If you enjoyed this episode, don't forget to follow, share, and give us a review. Thank you again for tuning in. We'll see you next time.